My name is Valentina Shakhnovich, and I'm a pediatric gastroenterologist and clinical pharmacologist at the Children's Mercy Kansas City. I'm here today to discuss the pediatric perspective on data analysis and interpretation with a focus on pharmacology trials, as that is my area of expertise. As a framework for today's discussion, we're going to build on the themes identified during the first workshop, namely, using age as the sole basis for analysis does not tell the full story. Data from highly selective trials cannot be extended to populations not included, which is why children of different ages need to be included in trials. And if our goal is to include children across the entire pediatric age range, then we're going to need to use different drug formulations in the same study, and that presents its own set of challenges when it comes to data analysis. To start off, age is not synonymous with maturity, and that is the biggest issue with the pediatric age binning system. When we look at the commonly used pediatric age bins of neonate, infant, child, adolescent, what this ignores is the tremendous amount of variability in organ maturity and organ function within each one of the bins. This is perhaps best illustrated in the neonates and infants, where there's tremendous amount of variability between infants born term versus preterm. The differences in their physiology due to prematurity can have significant implications for pharmacology trials. For example, when we examine what happens to the disposition or the behavior of one of the most commonly prescribed medications in pediatrics, acetaminophen, what we appreciate is when this drug is given as a suppository to children of different ages, we see very different systemic concentrations of the drug achieved, depending not so much on the age of the child when it comes to our neonatal bin right here, but it really has more to do with their age at birth and the extent of prematurity. This translates into our preterm infants having far higher concentration of the drug compared to full-term infants. As you can imagine, binning all of three subcategories together into our neonatal age bin is not going to tell an accurate story nor present the data in a meaningful way. When we take a look at the variability in the systemic drug concentrations across the child age bin, I think you can appreciate even that variability that we see in infants due to the different maturation of their GI tract translate into our child age bin as well. Why does this happen? Well, the easiest way to illustrate this is to show you a picture from a birthday party. These are 12-year-old girls, all born in the same month, in the same year. And I think you can see clearly that even though they're so close in chronological age, two of the girls are clearly prepubertal with very different organ maturation than the two girls who are post-pubertal. One way to get around this variability that's not captured in chronologic age is actually to base your pediatric age bins on maturity ratings. A commonly employed one is what's called the Tanner staging, which is a sexual maturity rating. The challenge here is that not all organs mature at the same rate. So for example, can we really extrapolate sexual maturity ratings to other organ systems? Perhaps the adult-like liver and kidneys are okay in a 12-year-old, but certainly the brain maturation of a 12-year-old, even though they may be adult-like on Tanner staging, is nowhere near adult level. So we really have to be careful about extrapolating to other organ systems and be thoughtful about it. Age is not synonymous with size, and this is another challenge in pediatrics that is particularly relevant to the 21st century. So we are living through the pediatric obesity epidemic. where 1 in 5 children in the United States currently meet body mass index criteria for obesity. Traditionally, these kids were excluded from clinical trials, but we can't afford to do that anymore, nor is it the right thing to do to exclude 20% of the patient population. So instead, we're going to think about how to best present pharmacology data for children who may be the same age as shown in this picture of 5th graders, but who are very different in size. You can imagine that giving 20 milligrams of a drug to this little guy here versus this young man here is going to result in very different drug concentrations simply due to size, nothing to do with age. One way to handle this and make comparisons valid across individuals of different sizes is to adjust the way that we present data and normalize it for the weight and the dose of the drug received. So instead of talking about drug concentrations in micrograms per liter, for example, it's not uncommon for us to convert the data into micrograms per liter per milligram per kilo of drug delivered in order really to make comparisons across different size groups. If we're going to include children of different ages in pharmacology trials, we're going to need to think about age-appropriate formulations. Generally speaking, it is not safe for children under 6 or 7 years of age to swallow solid formulations of drugs, so pills. Therefore, we rely on liquid formulations of medications for our younger age children, as well as other more creative formulations. The challenge is when employing 2 different formulations in the same pharmacology trial, it can be challenging to distinguish the age effect from formulation effect. For example, if we think about the drug cyclosporin, what we know about the drug is that the oral liquid form results in far greater systemic exposures in levels of the drug than the traditional adult formulation. So you can imagine that in a clinical trial with kids taking 2 different formulations where the younger kids get the liquid form, the investigators could erroneously conclude that there's an age effect on pharmacology when really they're looking at a formulation effect. To complicate matters more, most of you take medications probably with a glass of water. That is simply not true of younger children. Most children will take their medications with juice. As it turns out, children under 10 years of age account for the most consumption of apple juice in the nation. This matters, especially if you're relying on families to give the study drug at home, as they may be administering the drug with something other than water, which can have significant consequences on drug pharmacology. For example, this allergy medication, after a single dose administration, the levels achieved in the bloodstream are very different when the drug is given with water, say like in an older child, versus when it is given with apple juice, say like for a younger child. So again, you have to be careful about teasing out diet effect from age effect in pediatric studies. When we think about the diet across the pediatric age range, there's a tremendous amount of change during the first few years of life. So again, focusing on that neonate and infant pediatric age bin. Basically, a baby goes from an all liquid diet to a semi-solid and solid diet by 2 years of age. This matters because it influences the gastric emptying rate, which in turn will influence the absorption profile of any orally administered drug. For example, a baby in our infant category who's primarily on a breast milk or formula diet will have gastric emptying that is substantially different from an infant who has already transitioned to primarily a solid diet, resulting in potential pharmacology effects that may be attributed to age when in reality they're due to size or to diet. Whenever we're talking about pediatric studies, of course, we have to use age-appropriate equipment, and we have to be cognizant of age-appropriate cutoffs for things like vital signs and laboratory values for screening labs. So making sure your team is well informed and you know how to handle the data, especially when you see outliers. Since it isn't always feasible to conduct trials in children of different populations, or perhaps there are issues with enrollment rates, as you've heard earlier, data mining the electronic medical record represents a powerful tool to get around some of those issues with real life clinical trials. However, data mining comes with its own set of challenges. In order to be effective at mining your electronic medical record, you're going to want to partner with a bioinformatics specialist as well as a health expert in the area. I think if you look at this schematic, the steps that it takes from taking the raw data out of the EMR to even a stage where you can start data mining takes a lot of bioinformatics expertise. In addition to that, data pulled from the EMRs is dependent, at least on some part, on how it is coded with our ICD-10 diagnostic code criteria. This is a diagnostic criteria that's primarily developed for billing purposes, so it doesn't always capture the actual patient condition or the situation. To make things more complicated, there are currently over 72,000 ICD-10 codes. So you can imagine that myself and another gastroenterologist in my practice may code the same condition in very different ways, and that can affect the data on the back end when somebody is mining the EMR. The power of the EMR, in my opinion, really resides with the amount of heterogeneity you can capture in the electronic medical record. And heterogeneity is generalizability, because in order to generalize your data, you have to have all of the patients represented. So there's definitely a place for EMR to help us generate new knowledge that can be translated to the real world. Fortunately, we all live in the digital age, and even when clinical trials in a particular patient subset may not be feasible, there are now technological platforms such as physiologically based modeling computer tools that can aid with data simulation that can spark additional data analysis and interpretation. For example, in a PBPK model like this one, the physiochemical properties of a drug can be entered into a computer algorithm. The algorithm can be modified to account for pediatric physiology, for example, perhaps for the permeable gut we see in premature infants, or the small liver we see in smaller children. And that the computer then simulates the physiologic condition and generates a concentration time profile of how that drug is going to behave in that pediatric population. So this can be a very powerful tool, especially if certain populations aren't available, to help us augment our data analysis and interpretation. To summarize, there are significant limitations in age binning. Age is not synonymous with organ maturation, and age is not synonymous with size. If we're going to include children of different ages in pharmacology trials, we're going to need to account for teasing out age effect from formulation and diet effect. Generalizability is heterogeneity, meaning you have to include a wide variety of children to represent the entire spectrum of the patient population the drug is being designed for. And if you fail to account for age, size, and maturation-specific nuances in pediatric data, your data will not tell the full story.